So as James said, today our title is Infinium Assay, CNV Analysis using GenomeStudio and BlueFuse Multi software. And I will start our presentation today by saying that this webinar assumes that you have a working knowledge of the Infinium Assay and with using GenomeStudio. We have many resources if you are a beginner, but for the moment this webinar does assume. Our objectives today. First, we want to understand how Infinium genotyping assay data are used to analyze copy number. We'll talk about the importance of data clustering in Genome Studio for doing CNV analysis. We'll learn what B-Allele frequency and log-R ratio are and how they're used for CNV analysis. And finally, the relationship between allele patterns and copy number. We'll understand the differences in function and purpose between Genome Studio with cnvPartition and BlueFuse Multi. And finally, we'll demonstrate creating CNV analysis workspaces in both Genome Studio and BlueFuse Multi software. So to start, we're going to talk about how genotyping, clustering, and CNV analysis are related. Why are we using genotyping data for CNV analysis? And it's because using CNV genotype data, that is allele frequency, in combination with intensity data, offers advantages. CNV detection is confirmed using intensity data. This gives us an increased sensitivity for detecting mosaicism, and it provides us an ability to detect regions of loss of heterozygosity that have copy neutral. So first, the Infinium genotyping assay. The purpose of genotyping using Infinium arrays is to determine which variant is present at a given SNP locus for each chromosome in a sample. So we'll start by looking at the possible nucleotides for a SNP are written as AB in brackets. So for each allele on the chromosome, the variant can be either A or B. If we look at this table, we want to see what those possibilities are. This table is for non-homologous SNP, that means not AP or CG SNPs. So for an AG SNP, allele A is the A and allele B is G, and so on. Comparing to the possible genotype results are reported for each chromosome of a sample as either allele 1 and allele 2, or the terminology we'll use is allele A and allele B. So for biallelic SNPs, the possible combinations are AA, AB, and BB. So if we look at these possibilities for the actual genotype, for an AG SNP, the possibilities are AA, AG, and GG. And remember, this is for a sample, so this is the 2 chromosomes. Let's correlate that with the Infinium assay itself. This is a diagram of a cross-section of an Illumina BeadChip array. The beads that are embedded into the BeadChip are coated with oligos. Each bead is coated with an oligo specific for a specific SNP. In this diagram, we have 3 different SNPs. Written under the RS number is the possible genotypes. So the first one is the TG SNP, the middle one is TC, and the third one is AG. The sample genotypes, according to the colors that we see, are A's and T's are read in the red channel. So what we see on the right is both chromosome alleles for this sample have the A genotype. So this is AA, and it's all red. If we look at the left, G's and C's are read in the green channel. In this case, both alleles are the G genotype, and so this particular bead is all green. In the middle, this is a heterozygote, meaning one chromosome has the T and one chromosome has the C. This is pseudo-colored as yellow when we look at the scan images. So now let's correlate these colors with how we determine genotype. If we plot the red channel on the x-axis and the green channel on the y-axis, we can see three distinct populations correlating with the AA, AB, and BB genotypes. If we then take and plot the angle from the x-axis, and we're going to call that theta, and we see that at 0 degrees, the theta value is also 0, and the genotype is AA. At 45 degrees, we're going to say the theta value is 0.5, and that genotype is AB. At 90 degrees, the theta value is 1, and the genotype is BB. If we take that same graph, again, we're looking at theta value or angle from the x-axis, and then we add intensity. So we're going to call that value R, and the intensity is the intensity of the A channel, or the red channel, plus the B channel, or the green channel. If we then take that data, and instead of plotting red versus green, we plot theta versus R, or intensity. So we see here on the x-axis, we have the angle from the x-axis is theta, and on the y-axis, we have the intensity. More plainly, let's look at a SNP graph. So this is a graph for one SNP. Each dot represents a single sample, and each sample is plotted for both norm theta, or genotype, and the red and green signal intensity, or norm R. So, for a theta of 0, all signal is in the red channel, or A, so AA. A theta of 1 is all signal in the B channel, or BB, and a theta of 0.5 is the AB heterozygote. Then we look on the x-axis for normalized R, where the copy number is 2 at a normalized R value of 1. If we look at that more closely and we look at ideal copy cluster positions, we see genotype AA is a norm theta of 0 and an R of 1. AB is a norm theta of 0.5 and an R of 1. And BB is a norm theta of 1 and an R of 1. So let's look at a cluster graph, a SNP graph from Genome Studio. I want to point out to you the three different sections. So we have the red is AA, purple is AB, and blue is BB. And this is plotted norm theta on the x-axis, and again, norm R on the right axis. We see within each of those sections a shaded area. Within the shaded area is the oval, the cluster oval, and in the middle of that shaded area is the center that we call the centroid. So these are the features of a SNP graph in Genome Studio. I also want to point out to you that while this is very good clustering, the R values are not exactly the same between the different clusters, and that's perfectly normal. Sometimes the clusters will overlap or they're not well defined. So if a sample falls outside of one of the shaded areas, it's colored black in the SNP graph, and the genotype is not called for that sample. If the sample is within the shaded area but not within the oval, it will have the genotype called. Another anomaly can be if an assay returns a monomeric result. So we would expect that some SNPs, for some samples, will have a homozygous genotype that is either AA or AB, but it would be very unusual to see an AA genotype, just an AA. In general, cluster positions are consistent. So this is why we are concerned with having a good cluster file, which we'll talk about in a minute. And they should be, the clusters should be reproducible over large numbers of samples. So a graphical representation of that is if we run samples with a cluster file, then from batch to batch, we should get consistent data. And with the caveat that you're using high-quality genomic DNA. So we can save these positions by creating a cluster file. and then that cluster file can be used across projects. So this file saves the cluster positions for a panel of SNP assays. We use these cluster files to assign genotypes in Genome Studio. You can either create a cluster file within Genome Studio. It uses an algorithm called GenTrain to analyze the data points for each SNP assay. It defines those cluster positions, both the shading and the oval, and it assigns genotypes for each data point. And remember, these cluster files can be used for a particular sample type. They should not be used across sample types. You can also use a standard cluster file that's provided by Illumina for standard arrays. It is comprised of optimized cluster positions. That is, we evaluate thousands of samples over multiple batches, and we optimize the cluster positions, and we exclude poor-performing assays. When you use a standard cluster file, that means that you don't need to do the analysis of the cluster positions within Genome Studio, and that can really save time, especially for something like large projects. If you have a very large project, it's going to take a long time to calculate those cluster positions. So instead, you can use the standard file. For small projects, if you're trying to create a cluster file with less than 100 samples, if you don't have enough data points, those calculations for the cluster positions may be statistically insignificant. You can apply a cluster file, export it from a previous run, and calculate it with a large number of samples to use with the same panel of SNP assays and the same type of sample. Other considerations when using Genome Studio to cluster your data are, after you calculate the cluster positions, those positions need to be reviewed. And as I mentioned, anytime you create a cluster file, SNP assays should be screened for poor performance and then edited or excluded. Additionally, cluster positions may drift from the saved cluster positions, and the things that can affect that are laboratory conditions, including temperature calibration of equipment, pipette calibration, even laboratory technique, variable incubation time, and especially things like age of reagents and arrays. So we strongly discourage using expired reagents and arrays. And then finally, population effects. So if you're not using samples that are either the same sample type or from different populations, that will affect the clustering. A visual representation of this is, if we look at the SNP graph on the left, this is using the standard cluster file. The SNP graph on the right, we've clustered within Genome Studio, and you can see now that the cluster ovals and the samples have a much cleaner result. So we can see that the cluster fit can be better if you cluster on the data compared to using a standard cluster file. I'm not going to go much into creating cluster files at this point. We have several resources, including a recorded webinar, and these I've provided links here, and then two technical notes. One, Infinium genotyping data analysis, which provides step-by-step for creating a custom cluster file. And the second tech note is really more specific for copy number analysis. And it discusses the fact that it may be more important for you to use to create your own custom cluster file if you want to get the best copy number analysis. And so to reiterate, why is clustering important for CNV analysis? And it's because the copy number variation involves comparing the intensity data points from a sample in question with the expected data points from a normal sample. So let's look more closely at that. And by doing that, first we want to introduce the concept of B-Allele frequency. So again, if we look at the x-axis and norm theta and those theta values so we take those theta values and we assign them a B-Allele frequency of 0, 0.5, and 1, again, based on the genotypes of AA, AB, and BB. This B-Allele frequency is extrapolated based on the norm theta values. So if we look at the actual values along the x-axis for these three cluster positions, we see that the AA genotype is actually a theta of 0.04. The BB genotype is actually 0.57, and the BB genotype is actually 0.97. So what we want to do is determine the B-allele frequency of this sample in green. That data point has a norm theta value of 0.65. We take the difference between the distance of this particular sample and the AB cluster over the distance between this sample and the BB theta value. So looking at the map, that calculation is 0.2. So we know that this sample is 1/5 of the way between the B-allele frequency of 0.5 and the B-allele frequency of 1. So we assign B-allele frequency for this particular sample as 0.6. We can use the B-allele frequency plot in a utility in Genome Studio called the Illumina Genome Viewer to look for chromosomal anomalies. So if we, again, look at the SNP graph with the AA, AB, and BB genotypes, let's take that SNP graph and turn it 90 degrees counterclockwise. And again, we see the genotypes of AA, AB, and BB. Remember that a SNP graph is one SNP, and all of the dots are the samples that are in the project. If instead we take one sample and we plot all of the SNP genotypes for that one sample, we'll get a graph that looks like this. So again, we see the three nodes of AA, AB, and BB. So that's B-allele frequency. Another parameter that we use now is we're going to look at R, the norm R, called log-R ratio. And the way that's calculated is we look at R expected, and that is lines drawn between the centroids of the three clusters. R observed is where the actual data point is compared to those lines. So log-R ratio is the log of R observed over R expected. For data points falling on the blue line, the R observed is the same as R expected, and that equals 1, and the log-R ratio of 1 is 0. If the data point falls above the blue line, then R observed is greater than R expected, pardon me, and the log-R ratio will be greater than 0. If R observed is lower than the R expected, then that value will be less than 1, and the log-R ratio will be a negative number. If we then do the same thing that we did with B-allele frequency, so instead of the SNP graph with one SNP and multiple samples, we take and plot the log-R ratio value for every SNP for a particular sample. We get a graph. If nature were perfect and software were perfect, then every single sample in a SNP graph would have the exact R observed and R expected, and this graph would show us a very straight line with no deviation. But in fact, we do see that deviation, and that's what the number log-R ratio is for a given sample. If we take and compare B-allele frequency and log-R ratio, we're going to talk about slightly different values now. We're going to take the B-allele frequency, and instead of using 0, 0.5, and 1, we're now going to plot it as a percentage of B-alleles for that particular SNP for a sample. So if we take and extrapolate the data seen in the B-allele frequency graph in Genome Viewer, we can see that some SNPs are 0% because they're AA, some SNPs are 50% B-allele because they're AB, and some SNPs are 100% B-allele frequency because they're BB. If we then take the log-R ratio, we can do the same thing. So instead of assigning a norm R of 1, we're actually going to assign this a value of 2, meaning a copy number of 2. So we've taken the data from the log-R ratio graph and we've applied it. If we then look at a region of deletion, so there's been a deletion of a section of a chromosome. So now we only have one copy, as you'll see. So we no longer have that 50% B allele for this particular section. So we only have AA and AB, and that is 0% or 100% of B alleles. If we then look at the data in the log-R ratio, we see that the log-R ratio for this section is decreased, and we only have a copy number of 1. You will note that the actual decrease in log-R ratio isn't half of what it would be for copy number 2. Know that it's in fact, this decrease looks less than what you would expect. Let's look at an example of an amplification. We now have a duplication of a section of the chromosome. So instead of having just AA, AB, and BB, we have 2 heterozygote nodes. One is 33% B-allele frequency, and the other is 66% B-allele frequency, and those genotypes are AAB and ABB. We also see an increase in the log-R ratio, and we know that this is a copy number 3. So if we compile that, when we look at comparing B-allele frequency with log-R ratio, we can see in the first example that this is normal. It's a diploid, it has copy number 2, and we see the 3 expected nodes in B-allele frequency. The second example is a homozygous deletion, that is, it's just a deletion of 1 copy, and we see the corresponding homozygous phenotype in the B-allele frequency. For the third example, we see a duplication. That's a gain of 1 copy or 1 section of the chromosome, so that we see increased log-R ratio, and we see 4 nodes now in the genotype of the B-allele frequency. Finally, we see what also looks like a homozygous deletion, but in fact, if we look at log-R ratio, it has a normal copy number of 2. And what this means is that this is a copy-neutral loss of heterozygosity, meaning that this region of the chromosome has probably been duplicated to somewhere else. Using the comparison between the B-allele frequency or genotype and the log-R ratio or intensity, we can not only look at amplification and deletion, we can look at unbalanced aberration, aneuploidy, which is total loss, or mosaicism. And we'll talk about that in a minute. So we've been looking at data in Genome Studio. Let's look at it in BlueFuse Multi. Again, it's correlating log-R ratio, which is now on the top, versus B-allele frequency. BlueFuse Multi gives additional information in a pop-up window that says whether it's a gain or not, where the chromosome is, and what the size is. So again, we see that AAA, AAB, and ABB plus the BBB genotype. We can also see loss of heterozygosity. So the log-R ratio looks normal. The B-allele frequency is missing the AB track. And we can also use a feature in BlueFuse that shows all of the chromosomes, and it shows either red dots, which are a gain, green dots, which are a deletion, or this shaded region here, which indicates a copy-neutral loss of heterozygosity. Let's just look for a moment at mosaicism. So we can, again, compare B-allele frequency and log-R ratio and look at, in a population, sometimes there's the presence of more than one population of cells with different genotypes. So we can see different percentages of different populations. So in the B-allele frequency, we go from 0 to 100%, and in the log-R ratio, we can see the increase in that log-R ratio up to 100%, which shows 3 copies here at 100%. We can also look at deletions. So again, we have an increase in the split between the heterozygous nodes, except what we're seeing now is a decrease in log-R ratio, which represents a deletion of copy number. So how does Genome Studio and BlueFuse Multi detect and identify CNV? They both use B-allele frequency and log-R ratio, and they set parameters for analyzing. We look at variations from the expected pattern and fit those variations into models that correspond to different copy number states. Just as in clustering, software is not foolproof when detecting CNV regions, so each region should be analyzed separately and confirmed, either visually or with a secondary method. One other thing to consider with CNV detection is that we can get noisy data, especially in the log-R ratio. And you can see here in the depiction of log-R ratio in Genome Studio that this is very noisy. And then in BlueFuse Multi, what we see is many regions of deletion. And this is just due to noisy data rather than actual CNV detection. What can cause noisy data? Degraded starting material, because smaller fragments will be preferentially amplified over larger ones. Underloading, and for the same reason, can also cause wavy log-R ratio. If there's sample contamination, B-Allele frequency is affected, but log-R ratio is not. So in a sample contamination, you'll see multiple heterozygous nodes, just as you would with an amplification, but log-R ratio looks normal. Processing errors and expired BeadChips and reagents can also cause, pardon me. So instead of doing questions, I'm going to move on here, and at the end we'll have an opportunity for questions. So let's get into looking at how we look at copy number variation in Genome Studio. And the best way to do that is to do it using CNVPartition. If you're interested, there's another training on our website, and I've provided a link here. So what is CNVPartition? It's a plug-in software program that works with Genome Studio. It identifies regions of copy number variation, again, based on that R intensity and B-Allele frequency. And it uses the Illumina genome viewer, some representations of which we saw, to visualize copy number graphically, and then we'll also do that in table form. The requirements are to have the Genome Studio software and the CNVPartition plugin. So in order to do this analysis, we need a Genome Studio project that's already been created, and then we'll do a CNV analysis using CNVPartition. A note here that the better arrays for using CNVPartition for copy number detection is the larger density arrays such as the omni-family or specifically the cytosynth arrays. Low density arrays, because they have either rare variants or not sufficient SNPs, are not recommended for CNV analysis. So if we go to the Genome Studio support page, you'll see the plugins. You can download the CNVPartition plugin that will be on your computer, and you can run it, and we recommend that you install it to the default location suggested. Go to your project, open your project. Once in your project, under Analysis, choose CNV Analysis. You'll get a window that once you've installed the CNV plugin, it will be present in a pull-down. Choose that pull-down, and it will give you an option to set parameters such as the color of the copy number changes, several other variations, but mostly we recommend going with the default. You can then choose Calculate CNV Analysis. You'll get a window, a progress bar, and once that's completed, what you'll see then is a graphical representation of copy number. And we see here it's 4 different samples labeled across the top, and then for each chromosome that you see listed on the left-hand side. In addition to viewing that graphical representation, you can view CNV data in the full data table of Genome Studio. And how do we do that? First, we have to add a couple of columns to Genome Studio. Choose the column chooser in the taskbar, and then choose CNV value, CNV confidence in the hidden subcolumn, select show, and now in the data table, in the full data table, you'll see 2 columns. One is CNV value and the other is CNV confidence. So CNV confidence is calculated only when the CNV value is not normal. So if the CNV value is 2, and it's normal, you'll see that the CNV confidence value is blank. For an amplification, you will see a CNV confidence value as well as for a deletion. You can identify copy-neutral losses of heterozygosity by looking for those that have CNV value of 2 and CNV confidence with a value. So now we're going to go to the Illumina Genome Viewer in Genome Studio. So once we open up the Genome Viewer, we have a choice of choosing what samples we want to view. Then we need to choose 2 values. Again, B-Allele frequency and log-R ratio. Choose Add to Favorite and click OK. Once we do that, we can see a chromosome here. We can see our samples, but we still have to choose Update. So select Update. Now we can see all of the chromosomes for a given sample. But instead, it's productive to look at multiple samples and look at the B-Allele frequency and log-R ratio for those multiple samples. So we'll choose Chromosome Browser, and by doing that, we can now see the B-Allele frequency and the log-R ratio for each sample. And we can use the scroll bar on the left to go through those samples. So now let's get back to our CNV analysis. Under the View option, we then choose CNV analysis as bookmark. We get the same kind of screen here that gives us an option of what color to choose and what the intensity of those colors can be. And so once we choose our parameters, we can then click OK. And now what we see is for regions of copy number variation, we'll see shaded regions. And we see here kind of on the left, we see a green shaded region. And on the right, we see a blue shaded region. And if you hover over that region, then a box will pop up, and that will give you the bookmark type, meaning that it will tell you what the copy number is in the value. It will tell you the chromosome and the position. Now, let's see how we can get this data into a table format. So back to View, choose Bookmark Viewer, and now we can see this data in a table format. It has the same kinds of toolbar that Genome Studio does above the table. If we right-click on a row in the bookmark analysis, which I want you to note that it's sample ID, bookmark type, and then it's for each SNP, for each sample. So this is a pretty data-rich table here. But if you right-click on it and choose Go to Bookmark, it will then it will take you back to the Genome Viewer and show you the B-Allele frequency and log-R ratio plots. So we can export the data table, and I apologize for the animation here. We can export the table using the export function in Genome Studio. And again, we get this same list, but now as an Excel, as a CSV, comma separated value file. So in Genome Studio, we use cnvPartition to create a CNV analysis. We visualize the data in the full data table of Genome Studio and in the Illumina Genome Viewer, and then we use the bookmark analysis function to create a table and then export that table. So now let's look at CNV analysis in BlueFuse Multi. First, I want to compare BlueFuse to Genome Studio. So our target function for Genome Studio is research, while for BlueFuse, while it's not technically clinical, it is used in an environment where samples are being evaluated and data is being reported. Data is stored in Genome Studio as individual projects, whereas in BlueFuse, and you'll see this in a moment, All experiments are stored in a compiled database. In Genome Studio, genotyping is the primary function. In BlueFuse, CNV analysis is the primary function. We can and need to use and create cluster files in Genome Studio. BlueFuse relies on the cluster files created in Genome Studio. CNV detection can be performed in Genome Studio if you use the CNVPartition or just at a very high level approximating in the Illumina genome viewer. But in BlueFuse, that's the primary function. Input data files for Genome Studio can be either IDAT or GTC, and we'll discuss the GTC files in just a moment. BlueFuse uses only GTC files. To create a project in Genome Studio, a manifest is required, but the cluster file in the sample sheet is optional because you can always create a cluster file in Genome Studio. In BlueFuse, not only is the manifest required, but the cluster file is also required because BlueFuse does not perform clustering. You can use the BlueFuse version of a sample sheet called the batch import file as an option in BlueFuse. Genome Studio data can be created for any Infinium BeadChip in Genome Studio, but only certain BeadChips can be imported into BlueFuse, and those are CytoSNP12, CytoSNP850, and CarrierMap12. Some of the salient features of BlueFuse Multi. The first is the annotation database, which lists known CNV regions and associates those with disease if known. It links out to the UCSC Genome Browser and the database of genomic variants. Also, when you create a workspace in BlueFuse Multi and generate data, that data is stored in the BlueFuse Multi database. And you can query previous samples and see whether those samples have the same detected region that you are now seeing in your sample. Another good feature of BlueFuse Multi is that there's a hierarchy of sign-off. So once the data is analyzed, there's a sign-off feature, and then that makes that data reporting official. Another feature of BlueFuse is that you can do it in server mode, which means multiple people can have access to the data at the same time. So let's look at importing data into BlueFuse. First, we have to prepare the data, and if you remember, I said that BlueFuse requires GTC files or genotype call files. GTC files can be generated automatically on the iScan using autoconvert or separately using the Beeline software, and we'll look at that. So using the iScan, we can use a function called autoconvert, and this is just a brief overview of selecting that autoconvert function, and here's more instructions, but in fact what I have here is a link to the iScan system guide, and all of the instructions for switching on autoconvert start on page 32. The other way to produce GTC files is in a software called Beeline, and we're only going to talk about this particular function of Beeline today. You can learn more about Beeline, which does a facilitated data analysis. Again, Beeline cannot create a cluster file, and you would use a cluster file from Genome Studio. So it can convert IDAT to GTCs, and it also does genotype reporting. So for today, we want to talk about the function of convert IDAT to GTC. Oh, I want to mention that when you install Beeline, that you have to install both the Beeline software itself and an autoconvert software installer is required. So in Beeline, we're going to choose Convert IDAT to GTC files. We're going to navigate to the appropriate folder that has the IDAT. We need the manifest and the cluster file, and click Next, and then you'll see the progression of the creation of the GTC files. So now that we've created our GTC files, we want to import the data into BlueFuse Multi. There's a couple of ways to do that. First is the automated way, and we can use a template called the CytoSNP 850 planner, and that creates a batch import file, which is then used to import the data into BlueFuse Multi. Alternatively, you can create a batch import file and use that directly. There are two ways to manually import the data by sample. One is to use the full wizard, and one is to use the express wizard. So when we look at the automated method using the CytoSNP 850 planner template, it takes you through your experiment. You identify the samples, it takes you through the steps of the Infinium Assay, that those get logged, and then it brings the data into GenomeStudio, much like a LIMS system might do. So we enter this data, and it automatically creates the batch import file. This template is limited to 96 samples because of the 96 well plate format. That's how the batches are tracked. And it has built-in checks to make sure that you've identified the correct number of barcodes for the samples that you're saying you have. So this is the first tab of the planner, and it actually gives you instructions on that tab. And then as you progress through the different tabs, it gives you the different steps. You're going to insert your samples, you're going to add the barcodes, you'll add the 96 well plate format. So now your samples that you added in the barcodes will be figured into the barcode, into the 96 well plate template. The lab tracking form is used much the same way as when you run your samples. It has the reagents used and the operator, etc. It then creates the batch and then creates the batch import file. For additional information about this, I suggest visiting the BlueFuse Multi bead array walkthrough. And this is a feature that once you've opened up BlueFuse Multi, under Help, you'll see these walkthroughs. And there's a video specifically for arrays. It's a good, handy guide to have for importing data into BlueFuse Multi. Let's look for a moment at the format of the batch import file. It must have the structure where you cite the directory, the annotation database, and the data directory for each sample. And the data directory is where you've put all of the files that are needed to import those samples, such as the manifest and the cluster file, and then the data itself. So again, you can either create the batch import file using the planner, or you can create it directly. Once you have that batch import file, you'll go back to BlueFuse Multi, choose Batch, Batch Import, select the batch import file, select Open, and then you have a couple of choices. So while you're importing that data, you can select that it will start to process the data immediately upon import. And then you can also say, please poll for more data as is uploaded. Once you have finished making these choices, you select import, and then once finished, you need to cancel this screen. The other way to import data into BlueFuse Multi is manually. So when you're doing this, you're doing it sample by sample. You can use either the full wizard or the express wizard. And the full wizard allows you to enter associated data as part of your data entry. The express entry is just when you have the GTC file and the sample name, and that's it. And the same files are required as for batch import, so the manifest, the cluster file. And the same file validation is used to make sure you have the correct number of files for the samples that you have. So when you use the Express wizard here, you have just your sample ID. You need to make sure that you have the correct experiment type, and that's bead array. You need to browse to locate the GTC file. And once you've located the GTC files, the rest of these fields will be auto-populated. So the name of the BeadChip, the barcode, the position on the barcode or the location. And then you need to navigate to the cluster file and the manifest unless it's already been previously populated in the database, and then BlueFuse Multi will know for this BeadChip type, these are the files that I need. And the annotation database will default to the most recently used. Select Next, and that completes the wizard. And now you have your samples will be listed as you add each one of them sequentially. Once they're all added, you can then do batch process, and your samples will be processed. Instead of using the Express wizard to just enter the sample, you can use the full sample wizard to include additional information about the sample, type of sample, date it was run, etc. So using the full wizard, you add the sample ID, and that's the only one that's actually required. So you can save this, and then you can also go back and enter more data later using the Edit button. You can, if this is known, you can add the karyotype. And then again, it will take you to the section where you've got what the microarray is and the annotation database. And again, you can use this full sample wizard will open if you use the edit function. But once you have all this information and you click next, now you've got your data and it's been processed in BlueFuse. And again, I want to point out that BlueFuse uses both the B-Allele frequency and the log-R ratio, and it has algorithms that will look at the patterns and report that back in what the expected patterns will be. It can also report whether it's a gain or loss. It will give you the chromosome and position. It will also tell you, you can see whether it is a gain or a deletion by just looking at the data in BlueFuse. Another example here is the copy-neutral loss of heterozygosity that we saw previously. So to summarize importing data into BlueFuse Multi, it's easy to load it sample by sample, but the most efficient way is to do it with a batch import. And the most trackable way is to create that batch import file using the Cytosnip 850K planner. If all systems are on the network, this can be done in a minimum amount of time. And during batch import, process samples can be viewed and analyzed on a second computer when this is in server mode. So to summarize, we've looked at how Genome Studio and BlueFuse Multi apply very similar methods for utilizing the data of B-Allele frequency and log-R ratio to detect copy number variation. Genome Studio performs the data clustering and the genotype assignment. BlueFuse Multi, on the other hand, relies on the saved data cluster positions generated in Genome Studio. In BlueFuse Multi, data display formats and reports are dependent on the target user, so you can choose the way you're reporting your data, whether it's in a clinical environment or strictly research use only, and I say clinical in quotes. So we've added some resources to the end of our presentation. The Genome Studio and the BlueFuse Multi support pages are cited here. I also wanted to cite a publication, Mechanisms of Mosaicism, Chimerism, and Uniparental Disomy, which is a distinctive kind of copy-neutral loss of heterozygosity. And these are identified and discussed in this publication. It's a nice paper. So that's our presentation for today. Does anybody have any questions?